Before all of us, the genesis of Eukaryotes unilateralis. Deep in the rainforest lies the start of the New World, unbeknownst to the globe. The environment there is pouring with nutrients all throughout for a deadly organism. The air is its ideal habitat, a nice 94 to 95% humidity and 20 to 30 degrees Celsius. This moist environment houses many, one of them being Eukaryotes unilateralis, a fungal infection gathering steam to make its name known. This fungal infection is a deadly parasitic fungus. Its ideal geographical locations include pantropical or any warm temperature environment. The start could have been Australia, Thailand, South Carolina, Florida, or Japan, but it chose Brazil. This one summer night was the start of it all. The carpenter ant from the Camponotus family walks through the forest unaware that it's the main prey for this fungus. The ant is walking around at night trying to get food for its colony. As it's walking around, it notices fruiting bodies stemming from the ground. It goes closer, curiosity getting the best of it. Little did it know, curiosity would make its mind go crazy. Unknowingly, the fungus makes its move. Under the night sky, it settles on the ant's back. The ant continues on its journey, not knowing what just occurred. This would be the ant's last few moments of freedom, both physically and mentally. The fungus starts its takeover. The fungus, using enzymes like chintase, lipase, and protease, begins to break the ant's exoskeleton. While using mechanical pressure, the fungus attaches itself. It attaches to the dorsopronotum region right at the base of the ant's head. As the fungus makes its way in, it begins to synthesize compounds like sphingosine, hypoxanthine, and guanbionuric acid. These compounds aid the fungus in interfering with the ant's nervous system. The fungus invades the ant's cells and muscles. It starts altering organelles and amino acid balance. This total nervous system takeover is not changing the ant's DNA or RNA, but rather changing both CNS and PNS. As time goes on, the ant turns into a walking corpse, unable to control its own body. It starts to experience behavioral changes. It secludes itself from its colony, cuts off ties and communication, loses its daily rhythm and structure. It becomes hyperactive and staggered. All of this, yet the worst hasn't shown itself. Facing long, the ant is a walking corpse. It's a zombie ant. The ant is no longer controlled. It's not all the fungus. It's not conscious anymore, but it's still walking, eating, and breathing. It's still alive, but at what cost? The days are long and dreadful. One day, at solar noon, the ant sees a big tree. The height looks never ending. The road up looks daunting. Without a single ounce of control, the ant begins to climb up. This journey is known as treetop disease. One step at a time, the ant finally reaches a perfect atmosphere. The humidity, temperature, and available resources are screaming to the fungus, it's time. The ant makes its way onto a leaf, the start of the ant's end. The fungus forces the ant to bite onto the leaf. Controlling its muscles, it wraps the ant's leg around the leaf and forces it to stay. The ant experiences lockjaw because of the death grip. The ant is also now upside down, holding on for its dear life. The bite on the leaf's vein provides just enough nutrients to keep the ant's body alive. All that remains is housing for the fungus. To aid preservation, the fungus releases hyphae and invades the soft tissue. It fortifies the ant's exoskeleton and releases antibacterials that protect the ant from other microorganisms and other pathogenesis. The fungus does not want to lose its host, not just yet. Secondary metabolites like polyketides and naphthoquinone derivatives containing antibacterial and antifungal properties are what is protecting the ant's body. Now the reproductive stages can occur for the fungus. It's time for the fungus to expand. It can reproduce sexually or asexually. In anamorph, asexual reproduction, candidiospores called candidiophores are formed. The fungus goes through hirsutella or hymenostipes as these spores form at the stroma. In telomorph sexual reproduction, perithecia structures form. They're in the fungal stroma at the ant's head. The spores here are ascospores. Asexual reproduction tends to be the most common form due to how quick and easy it is. Telomorph may require a mate, anamorph doesn't. Now it's time to catch the next prey. In the end, the ant is nothing but a body with a stroma, a dark pigmented, weary mass of tissues containing perithecia, a flask-shaped structure with aci and ascospores. These spores explode, dropping the fungus contaminants onto the ground. The cycle will soon continue again. The fungal invasion, nervous system takeover, host protection, fungal reproduction, then spore explosion, a new start to the cycle. This time, the prey isn't an ant. The fungus is ready for a challenge. The next target, humans. Soon everyone will see what the fungus is truly capable of. The world must prepare, for this will be the last of us. Thank you.
Hi everyone, my name is Mia Nguyen. Today I'm sharing my project called Silent Invader: How Beetles and Fungi Are Killing Our Forests. This story blends microbiology and environmental science, showing how tiny fungi teamed up with beetles are transforming entire forests. So I'm gonna start with the art piece that I created for this project. On the left, you can see a healthy green pine forest. A mountain pine beetle climbs a tree, while purple fungal thread spread through the trunk and the roots. The circle show the fungal spore under a microscope, and on the right is the aftermath. The forest is brown and lifeless, beetles scatter the ground, and a harsh, really bright sun looms overhead, symbolizing the role of climate change. This drawing shows the invisible battles inside the trees and the larger impacts on our world. Let's meet the team of beetles and fungus. These two fungi here are Grossbenia clavigera and Ophiostoma montium. They can't move alone. They hitch rides on mountain pine beetles. When a beetle bores into a tree to lay eggs, it brings the fungi with it. The fungi block the tree's water flow, weaken its defenses, and make it easier for the beetle's larvae, the babies, to survive. And together they kill the tree. The relationship between the beetles and the fungi is a win-win relationship, and it is the perfect example of mutualism. The fungus needs the beetles to travel from tree to tree, and the beetle needs the fungus to weaken the tree so it can reproduce. Without one, the other can't thrive. Together, they form a deadly partnership, and that's why this outbreak is so damaging. So why does this matter to us? It is because we need our forests. Our forests are essential, they're not just beautiful. They give us oxygen, store carbon, and support wildlife. These beetles fungus pairs have destroyed millions of acres of pine forests in Canada and the US. Forests that took centuries to grow can die in just a few seasons. And when the tree dies, so does wildlife. For example, the Clark nutcracker loses its main food, the pine seed. Red squirrels, owl, woodpeckers also lose their nesting spot. Even elk, deer, bears, frogs, and bees are all affected. It's a chain reaction, and the dead forest increase wildfire and release more carbon, speeding up climate change. Talking about climate change, the climate change is making this worse. Warmer winter mean more beetles survive, and longer summers mean more time to spread. These beetles and fungi are now invading places very up north like Alberta and the Yukon. These areas was once protected by the cold. Let's talk about the microbiology of the fungi. These fungi are ascomyces or sac fungi. In the lab, they look like pale gray colonies. Under microscope, they form spores and branching threads. They're harmless to humans, but they're deadly to trees. And once they're inside, they overpower the tree defenses and block water transport, drying the tree from within. This project taught me that microbes, even tiny ones, can reshape our entire landscapes. It also shows how climate change is shifting nature balance. So, what can we do? I think the most important things we can do are support climate action to slow warming, protect forests through better land management. We can also educate ourselves about ecosystems and support research into forest and microbial health. Final thoughts. So I think even like a single spore, a beetles, a warming season, small things are changing the planet. And I hope this encourages you to see nature through a microbial lens and recognize just how deeply everything is connected. Thank you so much for listening. Have a wonderful day.
All right, hello everybody. My name's Claudia, and I'm doing my favorite microorganism on Batrachochytrium dendropatitis. However, it has pretty quickly become one of my least favorite microorganisms. While it's very interesting, I do not like the effects that it's had on the world, unfortunately. I'll be telling you guys about it through a silly little story about some frogs. Let's do spooky campfire stories. Yeah! No! I have one, but I'll have to warn you. This is more than just a story. It's real. I'm talking about Batrachochytrium dendropatitis. What? We'll call it BD for short. This strange organism is a fungus. It's part of the Chytridiomycota division, also known as chytrids. These are all unicellular, but they are one of only two chytrids that are parasitic to vertebrates. In fact, they're so unique that they're the only members of their genus. They're part of the order Rhizophidiales, family Incertaceidis. Yawn. This isn't even scary. Get to the good part. Calm down, guys. Did you miss the part where it's parasitic? To us? These little microorganisms start as little zoospores that can move around. They then turn into sporangium that can't move anymore. Then they get bigger and bigger, and then they pop, releasing even more zoospores to go around and infect even more frogs. In frogs, it can live in two ways. Zoospores can burrow deep into the skin and then grow into sporangium in the endobiotic cycle. These then pop and spread the zoospores even further. Or it can follow the epibiotic cycle, where they stay on the skin surface. However, this is less common. If you get infected, lots can happen. You'll stop eating, you'll be tired all the time, your skin will turn gray or white or tan or red, and you won't be able to turn yourself right side up. You'll get chytridiomycosis, which messes with the osmoregulatory of your skin, and eventually, you'll die. It lives by eating your keratinized tissues in your skin. They can spread through infected soil or water, or by frogs getting too close to each other, and then it can get even further as humans trade us around the world. They're still figuring out all the specifics, but they think other species like crawfish can carry it around and spread it. Some frogs can get it with no issue, but then it can get spread to a frog that's never seen it before and make them super sick because they don't have any immunity or resources against it. It was first seen in Australia in the near tropics, but people think it actually comes from Korea originally. They don't know the exact start. This is a heat map of all the places they found it so far from all the frogs that they have tested in the world. And now it's spread all over. It's been found in 86 countries and counting. These are just the ones that they've tested frogs at. It infects all different kinds of frogs. Did you know that we're in an extinction crisis right now? Up to 50% of the frog species are at risk. Uh-oh. BD can infect all amphibians, from frogs to toads to newts, salamanders, and caecilians. But frogs are the ones who are most impacted. Um, what was that? Your story sucks. I'm scared. Don't worry, guys. Domestic frogs can just get antifungal treatment. Yeah, but we're not domestic. I'm sure we'll be fine. We're hiding all the way out here. Dun, dun, dun. Thanks for listening, guys. That's all I have.
Hi, I'm Anna, and in this video, I'll be introducing you to Eustylagomatus, also known as corn smut. This microorganism is a plant pathogen, but also a cultural delicacy, and it inspired my art piece titled Beauty in the Blight. Here is a close-up photo of Eumatis. Eustylagomatis infects maize plants and causes tumor-like galls to grow on kernels, stalks, tassels, and leaves. While it's a problem for industrial farmers, in Mexican cuisine it's called huitlacoche and is considered a seasonal treat. Here is my painting. I used mixed media and it reflects this duality, something that is both a blight and a source of beauty. I used tissue paper and gouache paint to highlight the contrast between the vibrant corn and the dark fungal growths. Let's talk anatomy. So Eumatis is dimorphic, which means it grows as single-celled yeast under normal conditions, but switches to a filamentous form when infecting a host. This switch is essential for it to invade maize tissue and form galls. Phylogenetically, Eumatis is part of Basidiomycota phylum, class Eustachinomycetes, and order Eustachinelis. It's related to other smut fungi like Eustylago hordae, which infects barley. Its genome has about 6,500 genes, fully sequenced and used widely in fungal research. As for metabolism, it's facultative biotroph. As you may remember, this means that it performs aerobic respiration when oxygen is present, but can switch to fermentation in low oxygen environments. This flexibility helps it survive both in soil and inside plant tissue. Eumatis feeds on sugars and amino acids from the host plant. It thrives in warm, moist environments, especially during the rainy season. Its teliospores are thick-walled and can survive for years in the soil. It also interacts with humans in very different ways. Industrial farmers see it as a pest, but in Mexico, it's intentionally cultivated. People have eaten huitlacoche since pre-Hispanic times. It's rich in amino acids, fiber, and antioxidants. Here you can see them grilling it, and they can serve the whole corn um, entirely, or they can remove the pieces and put it into tacos or quesadillas. Here is a zoom of my painting skull, so you can see the texture. I wanted the calls to feel both grotesque and beautiful. The tissue paper captures the real-life softness of the fungus. The contrast of colors and the thick black outlines inspired by Aztec art frame the scene in a culturally respectful way. In conclusion, Eustylagomatus isn't just a crop disease, it's a culinary treasure, a model organism, and an artistic muse. Whether it's viewed through a microscope, on a plate, or as a symbol, it challenges us to look beyond appearances and find beauty in the unexpected. Thanks for watching.
My name is Brian. This is my term project for microbiology. The organism I chose is a Schistosoma hematobium. It is of the class Trematoda, order Strigatida, in the family Schistosomatidae. S. hematobium is a blood fluke or blood worm. It has a very unique life cycle. I find it pretty fascinating and terrifying. It's generally found in Africa and the Middle East. It is unique in that it has two sexes. It's unique that it has two hosts, and that it has a very, very interesting life cycle. So I'm going to show you what they look like right now, and then I'll kind of walk through the life cycle. But here is the male and the female. The male here is a larger one on the back. The female here is actually longer but smaller. and they pair up when they get down into the urinary tract and live in the visceral veins. They will pair up for extended period of times to release eggs. They have multiple suckers, one on the head and then one on the ventral. They are really not a problem living in the body as themselves. It is their eggs that's a problem. They create eggs that are going to look like this. What's got its terminal spine. The problem with these eggs is that they will go through the bladder wall and they are excreted through the urine there. It's in the little bladder there. That's my horrible job at a bladder, but they will continue out until they find fresh water. And once they find fresh water, this myocardium that here's in the middle that's fully developed will turn into this little ciliate myocardium, and they will like swim around in fresh water and look for a specific snail, which I find is wild. It's a Balinus snail. This is not really an accurate version of the snail, but just something I tried to make there. Those myocardium will enter into the snail. They will live off the snail's tissue and nutrients, and there they'll create sporocytes or sporocysts, and then they will just continue to create daughter sporocysts, and that they will eventually develop into these cercari, which actually look like this. They have these little tails on the end. They will leave the snail's body. They'll swim back out into the water, and then they will look again for a human host. Any human, this is a human leg here in the water. They'll find their way to somebody walking or playing in the water, and they will penetrate into the skin. At the time they penetrate the skin, they will lose the tail. They will travel through the bloodstream back up into the liver, where they will mature there in the liver, and then travel back down through the veins, through the portal vein, and end up back in the urinary tract, where they will look for another mate and start cycle over again. The biggest problem with these guys is that most of the eggs won't travel through the bladder. What they'll do is get stuck in the tissue there. Once they get stuck in the tissue, then it causes an immune response. Then you'll get inflammation, infection response, fever, nausea, headache, all that stuff. And then that's where you'll get a pathogenic response from them. So working on trying to get them eliminated, keep people out of the water, eliminate these snails, but it is an ongoing battle in those parts of the world. The thing I've tried to consider it like a xenomorph, and there's a picture of a xenomorph that I created there with the snails, but truly interesting animal. I've not seen one that would find a specific snail to go back into the human body. Wow. Thank you.
Giardia duodenalis, my least favorite microorganism. Van Leeuwenhoek, inventor of the microscope, was the first to study Giardia. He discovered this microorganism while doing a microscopic examination of his own diarrheal stool. Giardia duodenalis is a eukaryotic intestinal protozoan parasite. Giardia, which is the genus, are part of order Diplomonad. Diplomonads are flagellates with 2 nuclei found in anaerobic and microaerophilic environments. There are 6 species of Giardia: G. muris, seen in rodents; G. agilis, seen in amphibians; G. cetacea, and G. ardeae, seen in birds; G. microti, seen in prairie voles; and G. duodenalis, seen in humans and other mammals. Giardia duodenalis is a protozoan unicellular eukaryote. Protozoan organisms are morphologically classified into flagellates, ciliates, amoebae, and sporozoan. Giardia duodenalis is classified as a flagellated protozoan. Giardia duodenalis is a member of order Diplomonadida. Each microorganism in this order has 4 flagella, 2 nuclei, no mitochondria, and no Golgi complex. Diplomonads are also marked by presence of cysts in the organism's life cycle. and the ability to be free-living or parasitic. Diplomonads' two nuclei are identical in size and the amount of DNA molecules present in them, and both are transcriptionally active. Giardia duodenalis and other diplomonads are classified in supergroup Excavata along with sister group Parabasalids. Scientists have struggled for decades to determine how to categorize eukaryotic phylogeny. The current prevailing model contains 6 supergroups: Opisthecanta, Amoebozoa, Archaeplastida, Rhizaria, Chromalveolata, and Excavata, while the rest are commonly accepted as homogeneous groups. Chromalveolata and Excavata remain controversial groupings. Diplomonads were originally thought to be one of the earliest lineages of eukaryotes, but this view is no longer supported. Giardia duodenalis is an aerotolerant anaerobe that produces its energy via glucose fermentation, glycolysis. It has no mitochondria, instead possessing mitosomes, which are a reduced form of mitochondria. The degree to which these mitosomes participate in ATP synthesis is unclear. They are responsible for creation of FeS iron-sulfur clusters, which are important in its electron transport chain process. Giardia duodenalis exists in 2 different forms during its life cycle: cysts and trophozoites. Cysts are the resistant dormant stage of the organism's life cycle. They are responsible for transmission of giardiasis. Cysts are non-motile, oval-shaped, and have a 0.3 to 0.5 micrometer thick cyst wall with 2 more membranes inside, helping to protect the organism. When the organism is in its cyst stage, the flagella are pulled inside its cyst wall, and the adhesive disc, which helps the trophozoites attach to intestinal lining, is dissolved. The other form is the trophozoite, the vegetative, motile, replicating stage of the life cycle. Trophozoites are pear-shaped, with 8 flagella organized into pairs, basal bodies which generate these flagella, and an adhesive disc composed of microtubules. Metabolism varies based on life cycle state of Giardia duodenalis. The cyst is dormant and its metabolism is downregulated. Once the cyst is ingested, it becomes metabolically active and undergoes excitation. Excitation, the name for the transition from cyst to trophozoite, takes about 15 minutes and is triggered by gastric acid in the host stomach. Further down in the intestinal system, in the lower small intestine or upper larger intestine, encytation, transformation from trophozoite back into cyst, happens. The cyst is now environmentally stable, can survive in cold water for several weeks outside of a host, and is ready to be transmitted to a new host. Giardia duodenalis interacts with the human or mammal microbiota in a variety of ways. Giardiasis has been shown to interrupt the microbial biofilm structure, induce virulence in usually commensal bacteria, and interfere with the abundance and diversity of microorganisms present. Giardia duodenalis is transmitted by fecal-oral transmission. This can be person to person, animal to person, foodborne or waterborne. Humans need only ingest about 10 to 25 cysts for giardiasis intestinal infection to take hold. The incubation period after ingestion is about 1 to 2 weeks. Over 60% of infected individuals are asymptomatic, but those with symptoms can have diarrhea, foul-smelling stool, nausea, vomiting, cramping, flatulence, and weight loss. In regions of the world where giardiasis is endemic, there is some evidence of malnutrition and stunted growth in children resulting from giardiasis infection. Giardiasis is regularly seen in co-infection with other gut bacteria, viruses, and parasites. The adhesive disc's ability to attach to the intestinal epithelium, the flagellar motility of the organism, and the differentiation into cysts, which allows the organism to survive in stomach acid and in the external environment, are some of the major virulence factors of Giardia duodenalis and other Giardia species.